morning, dear students. Today we are going to learn the early development of Caenorhabditis elegans. What is this Caenorhabditis elegans? This Caenorhabditis elegans is a nematode worm which can grow up to a length of 1 millimeter long. It is a non-parasitic worm as well as it is free living. We can find it in soil. Then why this organism is considered as a model organism to study the early developmental process? There are a few reasons. First one, it's embryogenesis. That entire process is very rapid, so we can easily follow the entire process. Secondly, we can observe the entire process by keeping that organism even in a petri dish. That also we can accomplish. And this organism contain only a few cell types, different cell types. And predominant adult forms are hermaphrodites. These are a few reasons why we are considering this organism as a model organism to study early developmental process. Some more additional points, interesting points are here. The number of somatic cells in this organism is almost around 959. And the cell lineage is invariable between different individuals. What is cell lineage? Cell lineage means it is the history of each different type of cell, after which all process pathway it attain its final state. That is cell lineage, and that cell lineage is same between different individuals. They will not change. And one more interesting thing is that even if this worm is this much small, it has almost the same number of genes as that of human beings. That is about 18,000 to 20,000. Next, we are going to see the cleavage and axis formation in C. elegans. Axis means anterior, posterior axis, dorsal ventral axis, like that. The cleavage in C. elegans is rotational. What type of rotational? Rotational holoblastic cleavage. Holoblastic cleavage, we know that that cleavage will result equal sized cells. So, what is rotational holoblastic cleavage? In this type of cleavage, we can observe two planes of division. Which are those two planes of division? Meridional division, meridional plane of division, and equatorial plane of division. So in this rotational holoblastic cleavage, two planes of division we can observe. I will show you that. This is the zygote that formed after the fertilization in the case of C. elegans, and first division is meridional, and also it is happening asymmetrically. That means it is not dividing the cell equally, more equally. It is somewhat near to the posterior end. Okay, and as a result, two daughter cells will form, and one will be a founder cell and the other will be a stem cell. What is the difference between stem cell and a founder cell? Founder cells are those cells which will give rise to the cells of a specific type of tissues only, like hypodermis or pharynx or muscles, like that. Some specific tissue cells only will form from this founder cell. But stem cell is totipotent and it can give rise to almost any different types of cells. And okay, I will show you a picture. It will show how that division is proceeding. This is the first stage. As it divide, two cells are forming, stem cell and founder cell. We can see here this founder cell will become the future cells of hypodermis, neurons, pharynx and some other tissues. This stem cell again dividing and give rise to another stem cell and another founder cell, and that founder cell is directed to become the cells of some other tissues, and this P2 again dividing to give another stem cell and founder cell. In this way the entire process will proceed. Okay, I hope that is clear to you what is stem cell and what is founder cell. And next, these two cells need to divide. The founder cell always divide equatorially, but that stem cell always divide meridionally. Okay, and what will form as a result? As a result of division, that P1 stem cell divides and gives rise to one more stem cell, that is P2, and another founder cell. And a founder cell divides to give rise to the cells of some other specific tissues. And the founder cell that is formed in the second division is named as EMS, and the stem cell formed as a result of second division is P2, and it will proceed in that way. Okay, and the descendants of each founder cell. We need to know what each founder cell will form in future. In future, embryo, so that we can follow us that body of that organism is transparent. Its cuticle is transparent, so we can observe the descendants or the fate of each founder cell. And the time of that division, it occurs at a specific time and it will not vary between individuals, different individuals. And by the way of this rotation, holoblastic cleavage, how many cells are forming in newly hatched larvae? Around 558 cells are forming. And how we are naming the descendant cells? For example, I am showing the name of two cells, ABA. ABA means this is the cell formed as a division of this AB. This AB cell. As the AB cell divide, it gives rise to the ABA. And ABAL. ABAL means it is the daughter cell formed from ABA, and its position is towards left side of this ABA cell. And regarding this also, we can see the position. This is anterior to the AB parent cell. Is it clear? Okay. Next we are going to see the anterior-posterior axis formation. I hope that cleavage is clear to you. Anterior-posterior axis formation, how it is happening? What determines that anterior-posterior axis of C. elegans? It is determined by the elongated axis, which is the elongated axis. That defines the future anterior-posterior axis of the nematode's body. And which end will become the posterior and which end will become the anterior end, that is decided by the sperm's pronucleus that enters that egg during the time of fertilization. Okay. And as the sperm pronucleus enters the egg, we have learned that along with this centriole also, sperm centriole also entering the egg, and this centriole creates some cytoplasmic movement. It creates some cytoplasmic movements, and because of this cytoplasmic movement, what will happen? The sperm pronucleus moves and it occupies a place that is the nearest oblong end. Which is the nearest oblong end? that will be occupied by that sperm. And that end where the sperm occupied, that end become the posterior pole and the other end will become the anterior pole. Okay. So that cell division, cell specification means cell is specified to become a particular type. That's cell specification. And morphogenesis, all these are coordinated by the sperm centriole and several proteins. Protein means partitioning proteins. They determine that processes. And this sperm centriole, it is very critical in initiating the polarity, but it is not maintaining the polarity. And also, it is the sperm centriole itself is the main factor. It organizes, what organizes? Positioning of several maternal proteins at the future anterior and posterior ends of the egg. So these are some importance of the sperm's centriole. Next, I am going to tell you about the some important proteins that play a major role. Proteins means par proteins. I already told you that it play important role in determining the cell specification, morphogenesis, polarity, all those things. So how it determine the polarity? That's what I am going to explain to you. Mainly two different types of PAR and one MEX proteins are involved in here. This PAR3, this is the C. elegans egg. We can see that female pronucleus here. And before the time of fertilization, it's the cortex of the cytoplasm is filled with the PAR3 protein. And the inner side of the cytoplasm is covered by MEX5 protein. And while the time the sperm pronucleus along with the centriole enters the egg for fertilization, we know that because of the influence of centriole, some cytoplasmic movement happens, and as a result, that sperm moves to one of the, sorry, not sperm, sperm's pronucleus moves to one of the nearby oblong end and meet with the egg pronuclei to facilitate fertilization. And during that time came the significance of part 2 protein. What this part 2 protein is doing there at that time, it occupies the posterior cortical region of the cytoplasm by replacing part 3 and MEX5 protein. Okay, by that way now at this time the posterior most cortical region of the cytoplasm is occupied by only part 2 proteins, while anterior part is occupied by part 3 and MEX5 proteins. In that way, the anterior part and the posterior part are characterized. You can see that anterior part is characterized by part 3 in the cortex and the MEX5 in the interior cytoplasm. Posterior part is characterized by part 2 proteins in the cortex and a clear cytoplasm devoid of any MEX5 we can see in the posterior part. In that way, anterior and posterior characterization happen during that time. Okay. And after first cell division, that happens after fertilization and the formation of that zygote, the first formed founder cell, that is AB blastomere. That blastomere, we know that it is formed in the anterior end. So, it will have part 3 and MEX5 protein. And P cell or the P1 stem cell, it is formed from the posterior end. And as it is formed from the posterior end, it will have part 2 protein in most of its anterior end. That will be the result. Then what is the role of this PAR proteins? PAR proteins, partitioning proteins. It localizes so many P granules. I will tell you later what is this P granules. P granules, certain ribonuclear protein complexes, and this also specify the germ cells. Then what is this P granules? And the P granules are a collection of translation regulators that can be some proteins include RNA helicases, poly A polymerases, then some translation initiation factors. So, we are going to see what is the significance of P granules, how it specifies the germ cells. Shortly after fertilization, the randomly scattered P granules in the cytoplasm of the egg, it moves to one of its end, that is its posterior end, when it happens shortly after fertilization. Then the zygote undergoes first division. We know that it is a meridional division, and as a result, two daughter cells will form. One be a founder cell, and the other be the first stem cell is P1. And as that P granules is moved to that posterior most region of that zygote, this stem cell, first stem cell got this P granules. And again these two cells undergoes division and resulted from this first stem cell, again one more founder cell and one more stem cell will form. Its name be P2. Okay, and there also that P granules localized to the posterior most region. Okay, and the division process will proceed again and again, and always these P granules are localizing to the posterior most region of stem cells. I am just representing only the divisional process of stem cell here. And in all these stem cells we can see the P granules. And after some successive division, it will form the germ cells like egg or sperm of the adult tuber. In all these stages, in all these cells, we can see P granules. That means this P granules, whatever cells possess these P granules, that will specify the germ cells. And how these P granules are localized to the posterior region to get inside, to incorporate to this stem cell with the help of par proteins. Okay, and through with the help of what structure these P granules are localized, that is with the help of microfilaments. Not get confused that it is with the help of microtubules. Microtubules are not required for localizing P granules. What is the difference between microfilament and microtubules? These are two cytoskeletal element of cells. Difference is that its building blocks are different. Microfilaments is formed of tubulin proteins, and microtubules are formed by tubulin proteins, and microfilaments are formed of actin proteins. That is the difference. These two are two different cytoskeleton elements of the cell. So I hope that you understand how the anterior posterior axis is developing. With this I am going to finish today's section. In next section we can see how the dorsoventral axis and right left axis will develop. Okay, thank you. I'm an icon, man. Ain't my girl about a 10. I should fuck her in the Benz.